Today we are going to talk about the marssonina coronaria model. Marssonina is a disease that has been observed in apple orchards since a long time in China or in India, but in Europe it's observed mostly in the Alpine region since 2010. Initially, the disease was mainly detected in untreated or low-input orchards, and it can cause significant defoliation if it's not managed correctly. We'll select a weather station in Italy and open the marssonina model. In some countries, the inoculum can be both a sexual and an asexual form. In most countries it seems that no ascospores have been found on the soil. So probably there is only an asexual stage. Black acervuli become visible on the infected leaves, and those acervuli produce spores. The inoculum stays in the overwintering leaves, either in the leaves that stay on the trees or in the hail nets. We'll zoom out a little bit to see better what happens at the beginning of the season. The model is split in different graphs. At the bottom you can find a summary of the wetness period in light blue and the rain periods in dark blue that have been recorded by your weather station. This vertical blue bar here is the last data that was recorded by your weather station. So anything that is on the right side of this bar is based on weather forecast. The bottom graph is simulating the sporulation in the leaves. The model is starting mid-March at the date of green tip that has been entered by the user. You can see up here the date of green tip. The model starts with an arbitrary inoculum of spores that is produced by the acervuli in the overwintering leaves. The spores in the acervuli are modeled in brown. They need a period of surface wetness to prepare for spore liberation. Wetness records are in light blue in the bottom bar. We can see that when a wetness event is recorded, you can find free spores, modeled in yellow. Once that they are free in the water film, the free spores can be splash distributed to other parts of the trees. The longer and the heavier is the rain, the more spores are distributed on the vegetation. After the spores are splashed onto the trees, the middle graph is showing two processes: the germination, in white, and the infections, in red. Germination takes quite a long time. About 10 hours at 15 to 20 degrees for a light infection, and between 20 to 25 hours for a strong infection at the same temperatures. This explains why for the first rain events, some spores are being splashed and start to germinate, in white. But no infection, in red, occurs. Because the rain is short, and the wetness afterwards is short as well. The first real infection of the season only occurs around the 8th of May, due to multiple rain events and a long wetness period. The top graph is modeling the appearance of the symptoms with different stages. Let's zoom out for more clarity. Just after infection, the infected leaves are symptomless, in green, because the disease is incubating. After a couple of days, infected leaves start yellowing, modeled in yellow in the top graph. Incubation time is depending on temperature and relative humidity. After some time, necrotic lesions appear and the leaves start to drop, in brown. Until sometimes complete defoliation of the tree. From the moment leaves show first symptoms, a process of production of new acervuli is also started, because those symptoms can sporulate and create new conidias. This is modeled in black in the bottom graph. This process is driven by temperature and relative humidity. This is why infections tend to be stronger when the season is more advanced. Because you have not only the primary pool from the leaves of the previous years, but also the spores that come from the new lesions that have appeared on the leaves. Concerning the treatments, the most important graph is the middle one showing the infections. Targeted treatments are necessary. It is important to know that the weather conditions that are favorable for marssonina are also very favorable for secondary scab, fly speck or sooty blotch. So with one spray, you can try to target the three diseases together. In organic orchards, some fungicides, such as copper and lime sulfur, can be used preventively or as top treatment. Remember that marssonina comes with heavy and persistent rain, so the treatment should be repeated frequently enough to handle all the infections. If you have any question about this model, you can send us an email at support@rimpro.eu.